Enzymes are basically the small molecular workers of life. Every living being relies on enzymes. They basically govern every process that happens inside our bodies, but also in the bodies of every animal, every insect, every microbe. And the key fundamental that you would have to know about them, which is also nature's brilliance, you can think of a pearl necklace. And this necklace maybe consists of, let's say, 100 pearls. And for each pearl, you can choose one out of 20 different ones. So different color, different shape.
Hey there, biotech innovators. Welcome to the Smart Biotech Scientist podcast. I'm David Brühlmann, your host, and today we're diving into a fascinating world where enzymes meet cutting-edge artificial intelligence. Our guest is David Schönauer, the CEO of Aminoverse, who is revolutionizing enzyme engineering through AI. From discovering his passion for enzymes at university to leading a groundbreaking biotech startup, David's journey is one you won't want to miss. Grab your coffee and let's explore how the marriage of enzymes and AI is reshaping the future of biotechnology. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, David. Thank you for joining us today.
Yeah, great to be here, David.
I look forward to talking about how enzyme research meets AI. Let's start with the beginning. Can you take us back to your time at the university? What sparked your passion for enzymes during your studies?
Yeah, more than happy to do so. Enzymes very early on started to fascinate me, essentially because they are these small workers of nature, of life. They actually enable everything that we see around us. And on the one hand, of course, learning how this actually works and the fundamental principles behind it was, I think, what got me started. But then later on, it soon became clear that as soon as I learned more about this topic, I also realized that to a great extent, enzymes are already used in industry for the benefit of mankind and the planet. And it then became clear that I also wanted to contribute to this space and also, research in this fields and learning how research in that sense can be then translated to something more practical.
Yeah, that's exciting. And then actually very early on, actually still while being at university, you started becoming an entrepreneur. So can you tell us what inspired you to dive into leadership so early in your career?
The reason actually was maybe not the one you would think. It was more a flight from the lab bench. So for me, it was clear while doing the various internship courses and learning more and more how to become a scientist, my feeling was that the university or the system tries to form you into a potential future Nobel laureate winner. That's, I think, great for most of the scientists that go that route. But for me, it became clear that I do not really want to be the researcher at the bench or maybe the lab rat in that sense. I love to work with science and to think about it and solving, of course, the problems that come with it. That's the challenging part. But I definitely didn't feel the satisfaction that I thought I should feel. So I looked for other ways to still be connected to the science and contribute to this space, but while doing something different. And one way out there, and this was a bit sparked by also my professor at that time, who suggested me becoming actually an entrepreneur. And this was the first time I really started having this up on my list to consider as a potential future job, actually.
Yeah, that's amazing. And now you are an entrepreneur, you're CEO. So how did your experience with that university spin-off actually shape your vision for your current company, for Aminoverse?
It was essentially laying the groundwork there. So I was able to take over the helm as CEO. Obviously, this was then a lot of try and error, a lot of learning by doing. But then again, there's also no studies where you learn how to become a CEO. I mean, ultimately, it's a title. And by law, you have to have one in a company that carries that title. For me, it was more to be the person responsible for, of course, setting the direction, but at the same time being able to pursue the things that I wanted to do. And the company per se, I think, thrived. It was really growing for one person, then to 10 people over the next 7.5 years. So I think I really was able to implement and learn and also test the basics of entrepreneurship, keeping the money together, because there was no major venture capitalist who poured several million into the company. It was really counting every euro or every dollar, being sure when to make a purchase and when not to do one. And of course, also building a strong team, because without the colleagues, you cannot do anything.
So now I'm curious, since you're a CEO, how often do you still work at the bench?
Yeah, thankfully, as soon as I took over that position back in 2012, I never had to do this anymore for the business, at least. During the studies as well, of course. So since I did it in parallel, but after I completed or after I graduated, I only visit the lab when I want to see my colleagues, and that's good enough.
Let's talk about how enzyme research meets AI. But let's unpack this from the very beginning. Let's start in very simple terms, because can you just re-explain what enzymes are, and more importantly also, what is their importance in the biotechnology?
Yeah, more than happy to. So as I said initially, enzymes are basically the small molecular workers of life. Every living being relies on enzymes. They basically govern every process that happens inside our bodies, but also in the bodies of every animal, every insect, every microbe. And the key fundamental that you would have to know about them, which is also nature's brilliance, you can think of a pearl necklace. And this necklace maybe consists of, let's say, 100 pearls. And for each pearl, you can choose one out of 20 different ones. So different color, different shape. And then you can already imagine if you have 100 possible pearls and you can choose 20 different pearls each, you can make many different individual necklaces. And this is exactly what is also true for enzymes. So they are composed of amino acids. Everyone knows you need to eat protein, you need to eat the nutrients like amino acids and the building blocks of these workers, so to speak. And depending which combination of these amino acids you put together, you get different properties and different functions. So some enzymes are in the liver. They degrade, for example, alcohol if you drank a bit too much beer. Other enzymes, of course, then help build the structures or the muscles. Enzymes help to remove any clutter in the brain when you sleep at night, so you feel rested in the morning. So they fulfill numerous functions. And this is, of course, something that mankind about 50 years ago also discovered, that it might be possible to use actually these tools that nature invented for our benefit. And this is, for me, the fundamental principle of what biotechnology is. So it's using tools of nature, but in a more technological applied setting, that we can also benefit from that. So like an engineer, but they use, let's say, metal. And for me, this is like dead material. And I use now, or we use organic material. So enzymes, which basically is designed or invented by nature.
Well, obviously you can use enzyme in all kinds of different settings, in different reactions, in different places. Now, speaking about biotechnology, can you just highlight a few main areas or where enzyme are making a big difference?
Yeah, absolutely. So I think very clear examples are, for example, insulin or sugar diagnostics for diabetes patients. So this kind of detection of the sugar level is, for example, done by enzymes nowadays. You have also enzymes that were used for the mRNA manufacturing. So everyone remembers, I think, the COVID period we had a couple of years ago. This was a very innovative time, especially for the, let's say, vaccine or pharmaceutical field that were relying on RNA vaccines. And these are made actually by enzymes much more efficiently than by chemical methods. Enzymes can also be used for laundry detergents. So if you look at the washing detergent formulation or ingredients at home, you will definitely find enzymes on that. And other examples are also for nutrition. This is something that I think especially lactose intolerant people know. Everyone can buy lactases, and this is already a good sign. If something ends with an ASE, so ASE, it's like a lactase, you know it's an enzyme. And what is, of course, common to all of these examples, and there are many more, is that you are not able to just take an enzyme out of nature and use it in some of these applications. Why is that so? I usually like to give an example or compare this with a fever. Everyone knows fever is potentially dangerous, especially if it exceeds 40 degrees Celsius. And the reason for this is because at 42, 43 degrees Celsius, your enzymes die in your body, which means your entire metabolism, your basically the functions of your body will decline. That's why it's dangerous. At the same time, our normal body temperature is 37 degrees Celsius, which is not that far away. We are talking about 3, 4, 5 degrees. And our enzymes are really optimized by evolution for that. So we have 100% performance at 37 degrees, and we have zero performance at 43, 44. So we're talking about 5, 6 degrees difference. And this is life and death. So if you now think of an application like a washing machine, in the past, people were washing at 90 degrees or maybe 60. And nowadays, we can do it at room temperature or basically with cold water. And this is actually possible only because we added enzymes to the mix, or the respective companies did that. And this then required, obviously, a lot of, I would call it artificial evolution. So not done by nature, but by people like us, who then enabled these enzymes to also be fully active, so at 100% performance or even higher, in these non-native conditions, but in a very artificial condition, like a washing machine with all the detergent there and all the liquids and the clothes floating around.
Yeah, this leads me to the next question, because the universe of amino acids, or how you call it, the Aminoverse, I like that, is a vast space. And I read this on your website, more than 1 centillion possible combinations. So that's a big number. Can you tell us how big this number is?
No, actually, I mean, the comparison I usually do is that there are many, many more enzymes possible or existing than there are stars in the universe. Yeah, NASA estimates that there are 10 to the power of 24 stars in the universe. Maybe this is correct. I don't know. But if you then put this next to the number of different enzyme combinations where you have 10 to the power of 200, I don't even know how this number is called. But for me, this simply represents two things. On the one hand, this means there basically should be an enzyme for every application you can think of. But at the same time, it means it could be very, very challenging to actually really find that one needle in the haystack. And this is exactly where we come in, or why I decided to set up Aminoverse and use machine learning for that.
Can you tell us how you put the right enzyme into your customer's hand? Because as you have that many combinations, how do you do that?
Yeah, and this is exactly why I think will be the key to that. So it's clear that there should be the right enzyme out there. The question is, how do we find this? And what we then need is a way to see through these massive amounts of possible combinations and possibilities to end up with maybe not that one perfect match, but at least with a subset or a collection of very, very promising enzymes as candidates. And this is then exactly what we do. So in projects where a client approaches us, maybe they have maybe a chemical reaction in mind to make a pharmaceutical compound. We can then investigate what kind of reaction this really requires, what enzyme type this requires, and then basically we start going out there. We actually can access nowadays databases that contain more than 1 billion different enzyme structures and sequences, and basically can screen them with various tools to really filter them down, from 1 billion to maybe 100 relevant candidates that we then send to the lab and do the testing.
So what is the percentage between in silico versus in a wet lab?
Yeah, that's a very good question, because there is no definite answer to that. And this really depends on the actual problem at hand. Maybe as a rule of thumb, I would say the more delicate and complex chemistry you do, the more in silico you have. And the easier it is to test many variants in the lab. This is something that you would call high throughput screening. So we are talking testing several thousand, if not tens of thousands of different enzymes in the lab. Then you can rely a bit more towards the wet lab. But ultimately, and I think this is a key part of Aminoverse, we really consult our clients on finding the best balance between wet lab, the in silico part, and to some extent, of course, and also the machine learning part, because this is really augmenting both sides.
So how would this practically work? Let's say I want a specific enzyme, for instance, for a specific assay for my recombinant protein production. So I would come to you, tell what is the specific reaction we think is involved, and then how would you take it from there?
This is exactly all that we need. As a minimum requirement, we of course need to know where the client wants to go. So the reaction is the minimum. And then we can basically start using our tools. It's a platform that we call Enzyme for navigating all this possible enzyme sequences or navigating through that space and finding then most promising candidates. And this process is, you could see it like a funnel. So where you start with 1 billion sequences and then you start clustering them. You see what is the desired reaction, what kind of building blocks or features should these enzymes have to narrow down the selection. So if you were to categorize humans, you could say, okay, we want only people above the age of 65 and maybe only people that have been living in one country. And by these filter criteria, we can also apply similar aspects to the field of enzymes. And then we can narrow down the selection. And the final part will always be also a check in the lab, because as much as I'm a fan of nowadays in silico tools, biophysical simulations, it's amazing what the community as a whole achieved in that space. Now really, I think putting this on the next level with the field of machine learning. All of this is fantastic, but I'm also from the root, I'm still also a wet lab scientist, although I didn't like the bench that much. But it will never be, I think, replaced completely. And I think that's important to consider, to also know where the limits of these technologies are. And that's why I said, I think the most benefit that we bring to the table is how to strike the best balance. How much do I need to test in the lab to get some meaningful insights? How much can I rely on in silico tools or machine learning to do the job for me? And this is what our clients want. Yeah? They want an experienced partner who tells them right from wrong.
That wraps up part 1 of our conversation with David Schönauer. We've explored his fascinating journey from university researcher to biotech innovator and how he is bringing AI to the world of enzyme engineering. Join us in part 2 that will drop this Thursday, where we will dive into Aminoverse innovative approaches and the future of enzyme development. And before we go, are you looking to accelerate your biologics development while optimizing costs? Develop biologics better, faster, at the fraction of the cost with our fractional CTO services. Are you curious? If this is of interest, just book a free call with me. You'll find the links down in the show notes. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.